Hello, everyone. This is a new episode of our series on research talks and talking about the scientific method. Today, we will be talking about the osteoporosis, which is a very important global disease that affects a large population in the world. And for this purpose, we will have our PhD candidate, Yuliya Safarova, of the PhD in Science, Engineering, and Technology at Nazarbayev University, who will give us some hints, ideas on her research, and we will connect that with the research scientific method. Yuliya, welcome to our talk.
Hello, Liz. Nice to be here.
Great. I first want to learn more about you. What is your background? I know you are from Kostanay, Kazakhstan. And
Yep.
I want to know, how was your initiation in science? What was the motivation you had to start working on research?
Well, the research, that was an accident, actually. I mean, I didn't plan to be a researcher. I planned to be a doctor, like my parents and like my grandparents. So, and then there was an opportunity to enter the Bolashak scholarship program, and they were sending us to study abroad. And they were saying that that's going to be only biomedical science, not medical school. And then I agreed, and I did not have any idea that I'm going to be a researcher. So I just took a step into the unknown reality, and then I liked it. I mean, I've done my bachelor in New Zealand, and there were a really strong background for biomedical studies. And then once I came back, I was really planning to be in embryology field.
I see. And how did you connect? Did you connect with Nazarbayev University? What was the interest that came to you to come for the PhD in our program?
Oh, for PhD, yeah. The Nazarbayev University was a really unique opportunity to meet new professors and to be in my own country and do the research on the level of top universities, I would say. Yes, because we have really good laboratories and really good technical base that allows us to perform experiments on a good level.
And now let's try to go a little bit more into the topic of today's talk, which is the osteoporosis. Why osteoporosis? What is the importance of that disease?
Well, osteoporosis is age-associated disease. And currently there is a global aging of population. So the average lifetime and lifelong time is increasing. So we got more disease associated with age, and this is how, this is osteoporotic condition. And it really decreases their quality of life. Once you get the fracture, you are, it's really hard to heal osteoporotic fracture, and it often leads to disabilities.
I see. That brings us to the problem of your thesis. What is the problem you are trying to solve? What is the question you are trying to answer?
So the question I'm trying to answer is whether my approach with mesenchymal stem cells and osteophilic polymer will improve regeneration in osteoporotic bone condition. So that's the main problem I was trying to solve.
But being osteoporosis is a problem that has been quite a while in the environment. I mean, people know about that. What has been done so far, and what is the gap that you are trying to fill?
So there is a conventional therapy in osteoporotic condition, including bisphosphonate therapy. Those are drugs that decrease osteoclastic activity and improve bone density. And there is also hormone replacement therapy, and but it has several limitations, and including the risk of breast cancer development. Cell therapy that we are looking at can improve the regeneration rate and can help to escape those long time for rehabilitation.
Okay, but you are talking about that as a fact. But what was the hypothesis before you had? Because you are soon to defend. I know you have many results to present, but before you started doing the research, what was the hypothesis to make you think that you could improve the healing process?
The hypothesis was that this approach can improve regeneration rate in fracture in osteoporotic condition.
And what made you think that it was feasible?
Oh, I got it. So stem cell therapy is already on the market in some countries. You can do transplantation of own mesenchymal stem cells. However, lately it became evident that mesenchymal stem cells can possess a risk and can home to the areas of abnormal cell proliferation. So we functionalize those stem cells with osteophilic polymer to give it targeting potential. So the mesenchymal stem cells will home exclusively to the places of bone tissue and will regenerate there. And
and then your hypothesis when you started developing your thesis was clearly established. And how did you test the hypothesis? Did you do experimental work? Did you do numerical simulations? How was that?
So yes. We've done in vitro studies. The first step was to do in vitro studies and to show that this polymer possesses properties of binding to the cell membrane and binding to bone, having high affinity to bone. And we've tested how long the polymer stays on the cell surface. So it was all done in vitro on cell culture of mesenchymal stem cells. And afterwards, once we confirm that in cell culture it has this potential to bind cells, to drag them to the bone tissue and to decrease osteoplastic activity in vitro, then we transferred to in vivo studies. And for in vivo studies, we had a really good equipment called in vivo imaging computer tomographer. So with the help of this equipment, we could measure the bone density and perform in vivo experiments in rats for 24 weeks.
Okay, I know that part of your work has been developed with the help of partners, international partners. Could you please tell us briefly what was that part that you shared with international partners and what part was done at Nazarbayev University?
Yep, sure. So we have a connection and a really strong research collaboration with Carnegie Mellon University, and in particular laboratory of Alan Russell. So this is a laboratory that the chemical engineering laboratory, I would say, and they engineer the organic polymers. So the idea of polymer we developed together, and we've added the different functional groups to the backbone of the polymer together with our collaborators, and the polymer was synthesized on the base of the lab at Carnegie Mellon University.
Okay. And in local laboratories, what part was developed in connection with that?
All the in vitro studies, studies on cell culture were performed at National Laboratory Astana, and all the in vivo experiments were also performed at National Laboratory Astana, because we have really strong biological equipment related to cell culture and to in vivo analysis.
Great. Yuliya, before I ask you for the results, which is very important part of the conversation, I want to ask you about your daily life during the research part. How do you cope with the, lows and highs that we typically have in research? How was a typical week in which things didn't go as you expected? How was that energy? How do you motivate yourself to come forward and continue?
Well, thank you for this question. Actually, I don't know a single PhD student without any milestones arising, and so the main point is to overcome it, face reality, and take some actions to fix the problem. And then my main motivation is probably the main motivation is that I want to be a principal investigator. And so for this, I want to lead the research. I want to have a research team and to navigate them and to have my own research directions. And I think this is my main motivation.
Yuliya, now, if you can give us in a nutshell, what was the outcome of your thesis? Because you are defending very soon. Yep. What can you summarize is the most important outcome that you got in your thesis research?
Well, the most important outcome is that we've evaluated this approach in vivo, and we've performed short-term and long-term studies, and we've shown that cell therapy with mesenchymal stem cells and osteophilic polymer improves regeneration rate, and compared to control, in 1 month it regenerated 27%, and then it has a persistent effect for 6 months. So we performed the therapy for 1 month, but then the bone density remained the same for another 6 months. And that's a major outcome, because all the bisphosphonate therapy, it only lasts until you are taking it. And all the stem cell therapy, it also only efficient while like 1 or 2 weeks after you transplant the cell.
Excellent, interesting. Yuliya, in terms of publications, can you tell us what do you have as a publication from your thesis at this moment, and how important is the journal where you publish your papers?
The journal where I published my results, so that's Bioengineering, that's quite a new journal. It's somewhere around 7 years old, and it collects all the new ways in bioengineering. So somewhere it acts exactly where I am, at multidisciplinary field between biology, chemistry, stem cell research. So it's all together. And another paper I've published is a review in regenerative medicine, and I also have published in biogerontology.
Yuliya, now in terms of after your long trip journey during these past years for your PhD, what do you think are the major features you should develop as a researcher to be successful in science?
Well, I think the main abilities, I mean, that I got through my PhD journey, it's to be persistent, consistent, face reality. So those are a bit more psychological features. And yes, once I was entering the PhD, I was thinking only about my professional skills, but it turned out that PhD is more like a growing in psychological field, to grow as a person, to grow as a leader. Mm-hmm. And that's to grow as a research member. So, and not so much about the professional skills, but of course, we developed a lot of it.
Great, Yuliya. And thank you so much for this time that you gave us for this conversation about your research and your life. And I wish you the best of luck in your defense happening on spring 2021. Thank you.
Thank you. Thank you so much, Luis.